What is glycogen? To answer this, we're going to start with carbohydrates. When we eat carbs, our body breaks them down into what is called glucose. Glucose is the main source of energy, or fuel, for cells. When the cells are full of fuel, the body takes this extra energy and converts it to glycogen. So glycogen is a form of energy storage in the body. It is estimated that the body stores about 2,000 calories worth of energy as glycogen. It gets stored in mainly two places, in muscles and in the liver. Glycogen that is stored in the liver can be used by other organs and cells in the body. Glycogen that is stored in muscles is not shared, so it is used only by muscle cells. It is estimated that the liver will store about 400 calories of energy, and the muscles will store about 1,600 calories of energy. Now we're going to take a basic look at how this works. Let's say that Jack here is about to eat. His liver glycogen tank is 3/4 full, and his muscle glycogen tank is also 3/4 full. Jack eats his meal, and the carbs are broken down into glucose. Some of this glucose is sent by the liver into the bloodstream to cells throughout his body. The liver takes the extra glucose and converts it to glycogen and stores it for later use, filling up his liver glycogen tank. In between meals, when energy is needed, the liver breaks the glycogen down into glucose and releases it into the bloodstream. As you can see, the glycogen tank starts to empty until Jack eats again. One note here, fat can also be converted to energy to be used, and I'll cover that in another video. The process will be similar in the muscles. Jack's muscle glycogen tank was 3/4 full before his meal. After his meal, the tank is full. In between meals, Jack is moving around, causing his muscle glycogen tank to become depleted until he eats again.
Metabolism is just the flow of energy throughout the body. Energy enters our body when we eat food, and that food is then absorbed in 3 different forms. It can be absorbed as amino acids, so things that make up proteins, so you'd imagine meat would have a lot of amino acids. Or they can be absorbed as fats, so these are lipids or fatty acids, and so your greasy fried food is pretty rich in fats. Or they can be absorbed in carbohydrates, or I'll just write carbs here, which you have a lot of in ice cream or other sweet things. Each of these things deliver energy into your GI tract, your stomach and your intestines, which can then be absorbed and sent elsewhere for use. Now, carbohydrates are one of the main currencies for energy, so let's focus on that. And we'll do so by starting with glucose, which is the most basic form of carbohydrates. In fact, it's considered a simple sugar. Now, there are 2 main hormones that control the availability of glucose throughout the body, and they're in a constant tug of war with each other. One of them, which you've heard of probably, is called insulin. Insulin regulates the storage of glucose, as we'll talk more about in a minute, and the other guy on the end of the rope is a hormone called glucagon. Glucagon regulates the release of glucose from storage. And it's pretty important that we have enough glucose available in the blood because, for example, the brain uses about 120 grams of glucose per day. And that's a lot because it comes out to be about 60 to 70% of all the glucose that we eat in a day. But to put it in terms that I think you and I appreciate a little more, 120 grams of glucose comes out to be about 250 M&Ms in a single day. Now that's a lot of M&Ms. So you can see why it's really important to have enough glucose available for your essential organs to use. And thankfully we have these 2 hormones to help regulate the amount of glucose in our blood. So now let's take a look at how these hormones regulate the amount of glucose in our blood. And let's do that on this graph. So let's say this axis represents time, so over time we'll see some changes, and this axis over here, the y-axis, will represent the concentration of glucose in our blood. So that's the concentration of glucose. And most commonly that'll be represented in milligrams per deciliter. Milligrams per deciliter. Now, the body likes to keep the amount of glucose in the blood to be no lower than about 70 milligrams per deciliter and no higher than about 120 milligrams per deciliter. This is sort of the range that I would consider to be the sweet spot. Because if we go any higher than 120, then we end up having a condition that's called hyper meaning a lot of glycemia. Hyperglycemia, which just means a lot of glucose in the blood. If we have hyperglycemia for a long period of time, that can lead to what's referred to as eye, nerve, and kidney disease. Eye, nerve, and kidney disease. And we can go into a lot more detail about how this happens, but just understand that having a lot of glucose in your blood can cause changes to these structures to make them not work as well. And unfortunately, this is a fairly common problem, because another term for eye, nerve, and kidney disease is diabetes. And in fact, if you have a person who's been fasting overnight to come in for a blood test, and you notice that they have more than 126 milligrams per deciliter of glucose on 2 different occasions, that's grounds for diagnosis of diabetes. On the other hand, if we have very little glucose in our blood or not enough, that condition is referred to as hypoglycemia. Hypo meaning less or low, and then glycemia, of course, meaning glucose. And some of the things that you can start to notice if you're hypoglycemic is that you're tired, maybe you're lethargic. But if this persists, you can even go into a type of coma or even die from having too little glucose in your blood. And in most people, we start to notice that we're feeling hypoglycemic when we get below 40 milligrams per deciliter. Now usually, our body's pretty good about making sure that the level of glucose in our blood stays within the sweet spot or within this sweet range. And the way we accomplish this is through the hormones I just mentioned. So let's imagine that you eat at this point of time right here. And naturally, the level of glucose in your blood will rise because you've introduced more glucose into your system by eating it. Eventually, your body will notice that your glucose levels are rising and will counter that by releasing insulin to drive the amount of glucose in your blood down. And that's an important point because insulin decreases the blood glucose concentration by storing the glucose in another form. And we'll get into more detail about that in a second. The other thing that could happen is that you may have a decreasing amount of glucose in your blood, which, as I mentioned here, is not a good thing to have happen either. What your body does to counter that is release glucagon to increase the amount of glucose in your blood. And so it's important to remember here as well that glucagon will increase the serum or the blood concentration of glucose by releasing it from storage. So glucagon does the opposite. It releases glucose from storage. So now that we know how the release of glucagon and insulin can affect blood glucose levels, let's focus in and see how that happens. So let's start with insulin, and that does a number of things to glucose. But remember that at the end of the day, all we're doing is storing it. Just remember, insulin causes storage. So the first thing that insulin does to glucose is cause it to undergo a process known as glycolysis. Glycolysis, which you may have heard of before. It's an irreversible process. It's irreversible. All right, irreversible down here. Because it converts glucose into ATP, which is the most basic unit of energy that we use in the body. And that's an important distinction. ATP is energy to be used anywhere in the body. Okay, instead of storing the energy of glucose in ATP, insulin can cause glucose to undergo what's called glycogenesis. Glycogenesis, which just means the formation of glycogen. So glycogen. And glycogen is just a heavily branched polymer or molecule that has a whole bunch of glucose molecules stacked on top of it. And this is just energy to be stored in the short term in mainly the liver or muscle tissue, so mainly liver or muscle. And this is a reversible process, because once we make glycogen, we can break it down and release glucose as well. Finally, the last thing insulin can cause glucose to do is undergo lipogenesis. Lipogenesis, which I think you can use the suffix to infer here that we are producing lipids or fatty acids, so lipids or fatty acids. And this is an irreversible process. So this is irreversible, where we store glucose as lipid. And the key here is that we are taking the energy of glucose and we are storing it long-term in what's called adipose tissue. Adipose tissue, or just the fatty layers within our body. So adipose tissue. Now, what about glucagon? What are the processes it uses to release energy or glucose into the bloodstream? Let's put it this way. If we're releasing glucose into the bloodstream, my question is, what are we releasing it from? Well, the first thing we can release it from is glycogen, and we just talked about this. We can form glycogen using insulin, or if there's a lot of glucagon around, we can have what's called glycogenesis, which just means the breaking down or the cutting down of glycogen. Now, this is a reversible process because we can always go and take glucose to make glycogen again. The other thing we can release glucose energy from is, or rather I should say, are amino acids. Amino acids can undergo a process known as gluconeogenesis. So gluco meaning glucose, neo meaning anew, and then genesis meaning to create or the creation of. This is also a reversible process that will take amino acids, bunch them together with other things to convert them into glucose. Now finally, the last thing glucagon can do is to take fatty acid, so fatty acid or your lipid, and instead of converting it to glucose, glucagon will take the fatty acid and turn it into these things that are called ketone bodies. Ketone bodies. And it does so through a process known as keto, short for ketone, genesis, meaning to generate ketone bodies. Now this is an irreversible process, and it's kind of a funky thing that happens within the body because it's what we do when we're in our starvation mode, when we're not getting the right amount of nutrients for some reason or another. And the reason why this is sort of a last resort is because ketone bodies are very unique in that they are energy, forms of energy to be used only by the heart and brain. Ketone bodies don't really supply energy anywhere else, so that's why it's sort of a last-minute starvation mechanism to provide energy where it's most critically needed to help us survive. So you can sort of see here that there's a tug-of-war game that goes on between insulin and glucagon. In fact, insulin itself, when it's released into the blood, will inhibit the release of glucagon, which just goes to show you how opposite their end goals really are.
Now, a very simple premise that we've built our discussion of metabolism on is that we extract energy from food. And of course, this comes from the fact that we know that ATP is our body's main source of chemical energy, and the way we can produce this ATP is by breaking down nutrients such as glucose and fatty acids and proteins, which are all found in food. But of course, the question that you might wonder is, well, we don't constantly eat food, right? So how does our body produce a constant flow of ATP even though we only have food very intermittently? And of course, the answer to that question is that our body has evolved to store fuel as well. so that we're not reliant on an immediate influx of nutrients right after a meal. So in the remainder of the video, I actually want to go ahead and compare and contrast the three main types of fuel that our body has evolved to be able to store, and then touch on why one of these fuels is actually a much better storage fuel than the rest of them. So let's start off with glycogen. And glycogen is our body's way of storing carbohydrates. And essentially, it's just a long chain or a polymer of glucose molecules that are all attached to each other. And our body stores this mostly in the liver, but also there's some in our muscles as well. And if we were to tally up how much glycogen, how many grams of glycogen that we had, let's say in an average 70 kilogram male, an average healthy 70 kilogram male, we would calculate that there would be approximately 480 grams of glycogen. And just to give you some idea of how much energy we can extract from glycogen, we can extract approximately 4 kilocalories of energy per gram of glycogen. And kilocalories is just a unit of energy, and it's something that you might see on cereal boxes or any type of food really when you look at the nutrition label. And just to give you some perspective, the recommended average intake of energy for humans is somewhere around 2,000 kilocalories per day. And of course, this is a huge ballpark number. The exact number might fall above or below this and really depend on how old you are, what your sex is, as well as how active you are during the day. Now, a second type of fuel that our body stores is proteins. And remember that proteins are nothing more than a long chain of amino acids. And most of the protein in our body is in our muscles. And again, in an average 70 kilogram man, if we were to tally up how many grams of proteins, we would get around 6,000 grams. And again, similar to glycogen, we would be able to extract about 4 kilocalories of energy per gram of protein. Now finally, the third type of fuel that our body stores is in the form of fats. And these fats are actually stored up in specialized tissue in our body called adipose tissue. Now, if we were to tally up how many grams of adipose tissue a 70 kilogram man had, it would actually amount to a much higher amount than both proteins and glycogen combined. In fact, it's somewhere around 12,000 grams of fat in an average 70 kilogram healthy male. And moreover, unlike glycogen and protein, we can actually extract a lot more energy per gram of fat. In fact, that comes out to be somewhere around 9 kilocalories per gram of fat. Now, to put this in perspective, let's kind of do a fun, simple math problem here. Let's assume that this 70 kilogram guy requires an intake of 2,000 kilocalories per day. Now, if this is the case, let's kind of ask ourselves a very theoretical question, because of course it would be unhealthy to starve oneself, but if one didn't have an intake of food, how long would this man be able to survive on each type of fuel? So if we round this to about 500 grams of glycogen times 4 kilocalories per gram, that would actually amount to about 2,000 kilocalories that we could extract from glycogen. And so that would last him about a day, right? Now let's move down to protein. So proteins, we have about 6,000 grams in our body times 4 kilocalories per gram, which amounts to about 24,000 kilocalories divided by 2,000. That would last us about 12 days, right? So we're getting a little bit better. But now let's actually take a look at fats. We have substantially more amount of fats in the body than proteins and glycogen, right? So 12,000 times, let's round this up to 10 since we're just approximating anyways. And so if we multiply 12,000 times 10, that's about 120,000, right? Divided by 2,000, that's about 60 days that one could go, theoretically, of course, and probably may not even make it that far, but theoretically, we could make it 60 days on just our fat storage alone. Now, that's pretty impressive, I think. So now the question I want to answer is, why has fat become the major source of storage fuel in our body. And to do that, let's first actually remind ourselves what the chemical structure of fats are. So I've kind of, to save us some time, drawn out the chemical structure of a triacylglyceride. And I'll actually go ahead and write that out here. So triacylglyceride. Now, just as a point of clarification, I am using the word fat and triacylglyceride interchangeably, and that's because fat is just kind of an everyday term that we use to refer to the type of fat, the triacylglyceride, that we store in our body. And since we're talking about the chemical structure, it probably makes sense in this case to refer to it from its chemical name, which is triacylglyceride. And in fact, its chemical name tells us a lot about its structure, so let's take a look. So this triacyl refers to these three acyl groups, these acyl side chains. So what is an acyl side chain? So, an acyl is just a reference to a type of organic chemistry functional group. So there are some functional groups you might be familiar with, like hydroxyl groups or phosphate groups. And in this case, the acyl group is anything that has a carbon double bond oxygen attached to a long chain of carbons and hydrogens. And actually, I should say the chain doesn't have to be long. It just has to be some type of organic functional group. But in this case, the chain happens to be very, very long. And of course, I've kind of just gone ahead and drawn three acyl groups that have come pretty much randomly to my mind, because the idea here is that these chains can vary immensely depending on the type of triacylglyceride in our body, depending on the type of fats that we ingest. And so some of them might have single bonds, which we refer to as them being saturated with hydrogens, and some of them might have double bonds, in which case we refer to these triacylglycerides, or these side chains, as being unsaturated. So that's kind of just some nomenclature that you might see. And then finally, this glyceride refers to the backbone of this molecule, these kind of three carbons that are hanging out down here, and they also have a oxygen attach them as well. And of course, they link with these acyl side chains through this ester linkage that I'm kind of highlighting in green here. So that's the big picture of this molecule. Now, the reason why I think it's important to be familiar with the structure of a fat when trying to understand why fat is such a prominent type of storage fuel in our body is because now you can visually see where all of that energy, where all of that 9 kilocalories per gram of energy is coming from. Because looking at this molecule, you can see the bulk of it is formed by these long carbon-hydrogen chains. And these carbon-hydrogen chains are referred to as being very high energy because they have a lot of electrons kind of stored up in these bonds. And we know, if we remind ourselves back to kind of our general principle, is that if you have a reduced organic molecule like this, like we can see here, we are able to extract energy by oxidizing it in kind of subsequent steps. And this flow of electrons can be harnessed by something like the electron transport chain to allow us to produce ATP. So that's kind of my first point here, and I'll scroll down here and I'll go ahead and write that, which is that the triacylglyceride is a very energy-rich molecule. It has lots of these carbon-hydrogen bonds that can be oxidized to produce ATP. Now, a second reason why these triacylglycerides are such a good form of storage energy is because they're relatively chemically inert. So what I mean by this is that they're unlikely to react with other things in the body. And this is, of course, in contrast to things like glucose and proteins, which are quite polar. They have many polar functional groups, like hydroxyl groups, for example, and they can react with a lot of things in the aqueous environment of the body. But triacylglycerides are remember, they're not soluble in water. And just, remind yourself of, if you've ever made salad dressing, it's so hard to mix the oil and water together, right? And so because this isn't, going to dissolve in water, and it has a lot of these, carbon hydrogen chains that are considered not to be very reactive, it serves as a great form of storage energy because it won't, randomly react or be wasted in side reactions. A third reason why fats are good for energy storage is because they have no large or prominent functional role inside the body, in contrast to proteins, for example, which are used to make enzymes. And enzymes are, of course, of paramount importance in our body. And so we wouldn't want to rely too heavily on proteins because it would kind of be a conflict of interest for our body, right? Because if we use up too many proteins, we wouldn't be able to make enzymes. And that's why fats end up being kind of a good compromise, because we're able to kind of essentially just store up fats for one major purpose alone, which is to produce energy. Of course, that's a blanket statement. Fats are important in other ways as well, but largely speaking, their main role is to store energy. So I've touched on why triacylglycerides are a good form of energy storage and why proteins might not be such a good form of energy storage. But you might be wondering, what about carbohydrates? What about glycogen? Why didn't our body evolve to make glycogen the major storage fuel? And that brings me to my fourth point, which is that unlike proteins or carbohydrates, triacylglycerides, fats are very hydrophobic. And a benefit of being hydrophobic, as we kind of touched on earlier, so not only does that make it more inert chemically because it can't react with a lot of things in the water in the aqueous environment, but it also means that it won't be kind of weighed down by water. So you might be wondering, why is that beneficial? Why is it beneficial not to be weighed down by water? And to answer that question, we can actually just go ahead and do kind of a quick math problem here to kind of give some insight into that. So the math problem that I want to solve is how many grams of glycogen, including, I'm going to say this, including its water weight, because we know that all of these polar molecules attract some water, and that contributes, of course, to how much weight that they have. So how many grams of glycogen, including the water weight, will our body need, let's say, if we want to have the same number of calories? So I'm going to say if we're equal to the same number of calories, kilocalories, that the 12,000 grams of fat in the average 70 kilogram man that we talked about above can produce. So in order to answer this question, we have the information in the table above, but we also just need to know one more thing, which is how many grams of water are associated with each gram of glycogen. And the answer to that is there's about 3 grams of water weight, so say H2O weight, associated with 1 gram of glycogen, or protein actually for that matter. So I'll just put that in parentheses for our reference. All right, so to answer this question, we first need to find out how many kilocalories can 12,000 grams of fat produce. And that's simply 12,000 grams of fat, right, times, from the table above, I'll remind you, we said that for every gram of fat, we can burn about 9 kilocalories of energy. And because we're just trying to get a ballpark number, I'm going to go ahead and round that up to about 10 kilocalories, okay? And that is going to be equal to 120,000 kilocalories. So that's how many kilocalories we need to be able to produce with glycogen. Now, with glycogen, we know that for every 4 kilocalories, we're able to essentially utilize, or for every 4 kilocalories, we're able to get that out of 1 gram of glycogen, and that's without accounting for its water weight. But if we do account for its water weight, we know from this ratio above right here that for every gram of glycogen, there is 3 grams of water weight. I'll just put H2O here to remind us, okay? So our units are going to cancel out here, and all together, if we scroll down, we end up getting 120,000 divided by 4, ends up being 30,000 times 3, which ends up being a whopping 90,000 grams of glycogen that would weigh us down in order to produce the same number of calories as only 12,000 grams of fat. So nearly a 7 to 8 times fold difference in the amount of weight that our bodies would have to carry. And of course, this is not practical, right? Because we're talking about a 70 kilogram man, and here we have 90,000 grams of glycogen. That's equal to 90 kilograms, which is more than 100% of body weight just in glycogen alone. And so that's not practical because we haven't evolved a skeletal structure or enough muscle mass to be able to handle that extra weight. And so that's probably why fat has evolved to be the most prominent type of energy storage molecule in our body. So just to kind of summarize, if we return to our list up here, we notice that not only is fat itself, just by its structure, energy rich, it's also chemically inert. It plays no functional role, unlike proteins, which are important in enzymatic function. And because fats are very hydrophobic, we're able to pack a lot in our body without carrying any extra water weight.
In the last 2 videos, we've talked about how after a meal, you have a lot of glucose in your blood. There are 3 different cells in which your body is going to store that glucose so that you can use it over a long period of time. And the first of these cells is a muscle cell. The second kind of cell is a liver cell, which we also call a hepatocyte. Whenever you see hepato, you can think of liver. So a hepatocyte, which is a liver cell. And the third kind of cell where you're going to store the glucose is a fat cell, which is also called an adipocyte. Adipose tissue is fat. So an adipocyte is just a fat cell. So we've talked about how when you have all this glucose in the blood, it's going to get taken up into these cells. And we talked about the way that it's going to know to do that. And what was that again? That was insulin. So insulin is a hormone which your pancreas is going to release, and that hormone is going to tell these 3 cells to take up this glucose. In this video, I'd like to talk about exactly how these cells are going to store that glucose. So let's start with the muscle cell. The muscle cell kind of does the most simple thing. It takes a bunch of these glucose molecules and it links them together. So we can call this a glucose polymer. And actually, it's not just one chain. These glucose polymers branch out, and so they form this set of interlinking chains, which we call glycogen. And a full glycogen molecule actually ends up looking kind of like a little bit like a snowflake with all these branching series of glucose polymers. So that's the first way in which glucose will be stored. It'll be stored as glycogen in the muscle cells. So now how about the liver? How do you think the liver stores glucose? Well, you can answer that question because it's actually stored in exactly the same way as in the muscle cells. So the liver also connects the glucose molecules together to form glycogen. So how about the fat cells? How do they store the energy of the glucose? Well, they actually don't create glycogen like these first 2 guys. They use the energy of the glucose to make these things called fatty acids, which are kind of just long chains of carbon. And we're just going to draw them as a white line like that. And the adipocyte actually connects 3 of these fatty acids together to form a triglyceride. So it actually uses a glycerol molecule, which you can look up if you're interested, to connect 3 fatty acids together. And that's the form in which it stores the energy from the glucose. So this is what happens when you have a bunch of glucose in your blood after a meal and you want to store it. But now let's talk about how you actually make use of this energy later on. So a couple hours after your meal, your body is going to release glucagon, as we talked about in the last video. And glucagon is going to tell these 3 cells to release their energy. So let's talk about how that works. So here we have your blood vessel in this case. And let's start by talking about the hepatocyte. Now for the hepatocyte, it's kind of simple. It has all this glucose bundled up into glycogen. And what it's going to do is just to unbundle that glucose and put it back into the blood. So the liver is going to replenish some glucose in the blood by using up its glycogen. The adipocytes, so they're going to take these triglycerides and they're actually going to break them up into fatty acids and put them into circulation. and they can be used like that by some cells of the body for energy. And finally, the muscle cells do something a little different. The reason why we didn't start with them is because they actually don't put the glucose back into circulation. Muscle cells are selfish, and when they get the glucose, they make glycogen, but they make it for themselves later on. So what the muscles will do when they need the energy is to break the glycogen down into glucose and then use that glucose themselves for energy. And again, all these reactions are essentially told to occur by glucagon, which is released by your pancreas when you don't have enough glucose in the blood. Now, before we finish, I thought it might be fun to talk about exactly how much of this glycogen these cells can store. So the liver can store enough glycogen so that that glycogen will make up about 5% of the liver's mass. And that comes out to about 300 kilocalories of energy. Meanwhile, the muscles can produce enough glycogen to make up about 1 to 2% of their mass. But as you might guess, you have a lot more muscle in your body than you have liver. And so that actually amounts to more than the liver. It amounts to about 1,200 kilocalories. And it's worth noting, this unit kilocalorie is the same as the calorie number that's given in the nutritional details of foods. But as we said, the energy that your muscles store can only be used for the muscle. So the rest of your body has to get by on the glycogen of your liver. But 300 calories really isn't that much. It only lasts you a couple of hours. So we see that it's actually quite important that we have this other way of storing energy, these fatty acids, because without it, we'd run out pretty quickly.
Hey everyone, in this lesson we're going to talk about glycogen synthesis, also known as glycogenesis. So the first thing we're going to get into is what glycogen is, how it is actually synthesized, what are the enzymes involved in glycogen synthesis. Then we're going to talk about where glycogen is stored in the body, and then finally we're going to talk about what the role of glycogen is in our bodies. So to begin, here is a picture demonstration of what glycogen is actually looks like. And as you can see, it's a very complex branching structure with a core enzyme. And we'll get into what that core enzyme is in a moment. Now, glycogen, if you break down the word glycogen, it's actually the prefix glyco means glucose, and then the suffix gen means producing. So it's glucose producing. Now, in fact, glycogen is simply just a large polymer of glucose monomers. And glucose monomers can be arranged in a linear fashion with linkages of alpha-1,4 linkages. And it is also a branching structure, as I mentioned before, and the branch points are actually alpha-1,6 linkages. Now, the carbons are all numbered in a glucose molecule, and again, as I mentioned before, the linear linkages are 1,4, and the branching is alpha-1,6. Another important thing to note is that glycogen has branching points at every 8 to 12 glucose monomers, which is important because it distinguishes glycogen from similar compounds such as amylopectin. Now, glycogen, because of the word glucose producing, it is in fact a rapid source of glucose, and that's the key about glycogen, is actually a rapid source of glucose, and it is highly important because it actually maintains blood glucose levels. And during prolonged fasting, glycogen stores in our body typically only last about 24 hours or a little less than 24 hours. Now, I mentioned before there's a enzyme in the core of the glycogen. Now, this enzyme is called glycogenin, and I like to call this enzyme the catalytic core, and it's because it actually is an initiating enzyme. It itself begins the process of glycogen synthesis, and it does this by actually autoglycosylating itself, and it'll actually produce or attach chains of about 10 to 20 glucose monomers to itself. And that's how this whole glycogen synthesis process actually begins. So stepping back, we start with the glucose. Now, when a glucose enters a cell, it can actually be phosphorylated by hexokinase or glucokinase, as we've learned before in other lessons, to glucose 6-phosphate. Now, I always like to call glucose 6-phosphate the hub of many different signaling pathways. It, in fact, can be used for several different purposes. Now, the first thing it can do is actually it can go back and be dephosphorylated to glucose by glucose 6-phosphatase if glucose is needed. However, it can also go into other pathways. One of them, glucose 6-phosphate, can be processed to fructose 6-phosphate and then used for glycolysis, for energy production. And the whole topic of this video, though, is that if it's not used for any of these pathways, it can be used for glycogen synthesis by being shunted or processed into glucose 1-phosphate for glycogen synthesis. So if the glucose 6-phosphate is used for glycogen synthesis, how does that actually happen? Well, the first thing that happens is that glucose 6-phosphate is acted on by the enzyme phosphoglucomutase to form glucose 1-phosphate. And pretty much this enzyme simply just shifts the phosphate group from carbon 6 onto carbon 1 to form glucose 1-phosphate. Now, it's important to realize that this step is a reversible step, which means that glucose 1-phosphate can be reprocessed into glucose 6-phosphate if the cell requires free glucose. And this is important during glycogenolysis, which I will talk about in another video. Nevertheless, if glucose 1-phosphate is going to be used for glycogen synthesis, what's the next step? Well, the next step is actually glucose 1-phosphate being acted on by UDP glucose pyrophosphorylase. Now, this is a very important enzyme. It takes UTP and it activates glucose. This is what we call the activation step. UTP will actually attach a uridine moiety to the glucose to form UDP glucose, the active form. So once we have UDP glucose, what is the next step? Well, the next step is glycogen synthase. Now, this is a very important enzyme, and what it does is it actually will remove the UDP portion off of UDP glucose and add the glucose to the to actually a non-reducing end of a glycogen chain. So what it does is it'll actually just pop the UDP portion off of the UDP glucose, and it will actually pop the glucose monomer onto a non-reducing end of a glycogen chain of glucose monomers. And it does this again in an alpha-1,4 linkage. It forms an alpha-1,4 linkage. So once we have a very long glycogen chain, where do the branching points come from? I mentioned glycogen synthase attaches glucose to glucose with an alpha-1,4 linkage, but I mentioned before branching points are alpha-1,6. So how does that happen? Well, if we can imagine a glycogen chain, a very long glycogen chain with a glycogenin core, how does the branching points, or where do the branching points come from? Well, simply, there is something called a branching enzyme. Now, the branching enzyme, what it does is it'll actually remove 6 to 8 glucose molecules off of the terminal end of the long chain. So what it'll do is it'll actually chop off the last 6 to 8 glucose molecules off a long glycogen chain, and then it'll actually attach that chain it chopped off back onto another chain of glycogen with an alpha-1,6 linkage. And that's how the branching enzyme actually produces branch points. So that's where we get the very branching structure of glycogen. And this is also very important to point out that with the more branch points and the more free terminal ends that glycogen actually has, it actually speeds up the rapid release of glucose. Now, instead of having, in our example, instead of having one chain with one free glucose at the end where one can be removed at a time, now we have 2 points where 2 glucoses can be removed at a time. So the branching of glycogen actually speeds up the process or allows the cell to access more glucose quicker and faster. So that's why it is actually a rapid source of glucose. So we've talked about the production or the synthesis of glycogen, but we never talked about where this is actually happening and where it's stored. Well, as I show you here, the two major sites of storage in our body for glycogen is the liver and the muscle. Now, with the liver, the glycogen is actually stored in the cytoplasm of the hepatocytes, and its weight is about 100 grams. And the role of glycogen in the liver is pretty much to maintain blood glucose levels. That's its main role when it's actually stored and used in the liver. And again, it's used during fasting. And this is this storage of, or this reservoir of glycogen usually only lasts for about 24 hours or a little bit less than 24 hours. And its main role is just to maintain blood glucose levels. Now, with a muscle, again, if glycogen is produced in the muscle, it's actually stored in the cytoplasm of the muscle. And in totality, in muscle, glycogen will actually come out to be about 400 grams of muscle mass is actually glycogen. And its role in the muscle is actually an energy source, and it's actually utilized during exercise. So when you are exercising, when you are having acute periods of exercise, you begin to use muscle glycogen stores, and that is used simply for rapid glucose, a rapid source of glucose, and a rapid source of energy.
The word gluco means glucose, neo means new, and the word genesis refers to the production. Under normal circumstances, the carbohydrates are broken down in the human body by various enzymes into glucose. But this is just a breakdown. Under special circumstances, substances like fat, amino acids, pyruvate are converted into glucose. Thus the production of glucose from new sources. Thus, gluconeogenesis can be defined as the synthesis of glucose from non-carbohydrate sources. The average person consumes food many times in a day, which ultimately gets converted into glucose, and this maintains a blood glucose levels between 100 to 200 milligrams per deciliter throughout the day. The excess of the glucose gets converted to glycogen, which is stored in the liver, and this is used later when the blood glucose levels fall. Under a fasting state, the blood glucose levels start falling, and excess of the glycogen, which is about 100 grams, gets converted into glucose, which maintains a blood glucose level. But this glycogen reserve soon gets depleted and the blood glucose levels start falling. But some tissues in the human body, like the nervous tissue and the red blood cells, can only utilize glucose as a source of energy. So substances like fat, amino acids, and pyruvate get converted into glucose by gluconeogenesis, which maintains the blood glucose levels. About 50 to 60% of the gluconeogenesis takes place in the liver, 40% of this process takes place in the kidneys, and some minor amount of gluconeogenesis also takes place in the intestines. The metabolic reaction involved in the process of gluconeogenesis involve glycolysis, Krebs cycle, and some other reactions. So we consume food in a lot of complex carbohydrate forms throughout the day, which through our intestines gets converted to glucose. Glucose is a 6-carbon compound, and if you have studied the video on glycolysis, you know that the glucose is converted to 2 molecules of pyruvate, which is a 3-carbon compound. This happens through a series of 10 reactions known as glycolysis. Out of this, 3 reactions are irreversible. It is very important to understand the significance of these 3 irreversible reactions. These reactions are catalyzed by specific enzymes which are upregulated only when the concentration of glucose is high, so that the excess of glucose gets converted to pyruvate. This prevents excess concentration of blood glucose. But these three irreversible reactions can be reversed using specific enzymes which are upregulated only when there is a deficiency of glucose in the blood. These reactions are upregulated during special circumstances like fasting. During fasting, this pathway can run in complete opposite direction so that the pyruvate gets converted to glucose. Other substances like propionate are first converted to intermediates of Krebs cycle, which then get converted to pyruvate. And substances like lactate are first converted to pyruvate directly, which convert to glucose later. So here is an overview about the process of glycolysis. Glucose is converted to glucose 6-phosphate by the enzyme glucokinase. The glucose 6-phosphate is then isomerized to fructose 6-phosphate. Then the enzyme phosphofructokinase adds another phosphate at the first position and converts it into fructose 1,6-bisphosphate. And through a series of reversible reactions, fructose 1,6-bisphosphate is converted to phosphoenol pyruvate. And through a last irreversible reaction, phosphoenol pyruvate is converted to pyruvate by the enzyme phosphofructokinase. So the first reaction, which is irreversible and we have to talk about, is the conversion of pyruvate back to phosphoenol pyruvate. For this, pyruvate first travels to the mitochondria through specific carrier proteins. In the mitochondria, pyruvate is converted to oxaloacetate by the enzyme pyruvate carboxylase. The enzymes in the green are the gluconeogenic enzymes which are expressed during fasting. In this reaction, carbon dioxide is used and ATP is also used. Oxaloacetate is then transferred back to the cytoplasm, which through the help of the enzyme phosphoenol pyruvate carboxykinase gets converted to phosphoenol pyruvate. In this reaction, GTP acts as the electron donor and carbon dioxide is liberated back. Phosphoenol pyruvate then is converted to fructose 1,6-bisphosphate since these reactions are reversible. The next irreversible reaction is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. This is brought about by the enzyme fructose 1,6-bisphosphatase. And the last irreversible reaction is the conversion of glucose 6-phosphate back to glucose, which is brought about by the enzyme glucose 6-phosphatase. Phosphatases are the enzymes which break the phosphate bond. Now other substances like propionate, lactate and glycerol also are converted to glucose through specific pathways. Let's first talk about propionate. Propionate is first converted to succinyl-CoA. And if you have watched the video on Krebs cycle, you know that the succinyl-CoA is an intermediate in the Krebs cycle which get converted to oxaloacetate, and then oxaloacetate is converted to phosphoenol pyruvate. Substance like lactate is converted directly into pyruvate by the enzyme lactate dehydrogenase. And glycerol, which is derived through the fat tissue, is first converted to glycerol 3-phosphate, which is then converted to dihydroxyacetone phosphate. And dihydroxyacetone phosphate can be converted to fructose 1,6-bisphosphate through the reactions of glycolysis.
What I think is pretty fascinating is that our body is able to maintain a very narrow and constant range of blood glucose in our body. So notably about 60 to 150 milligrams of glucose per deciliter of blood. And it's not important that you know this exact number, but what I think is significant is that in contrast to something like free fatty acids, for example, which we'll talk about in fatty acid metabolism, fatty acids can range almost tenfold depending on the needs of the body. So they can either be really, really high or really, really low. But glucose always stays within a very constant range. I'm going to say blood glucose level here. And it's important that this is a very constant range, of course, because there are some tissues in our body, such as our brain and some of the cells in our eyes and our kidneys and even our red blood cells that rely on glucose nearly exclusively to produce ATP. So remember, once glucose is in the blood, it can be used by any of the cells in our body through process of cellular respiration to produce ATP. So remember, the three big steps of cellular respiration are first glycolysis, the breakdown of glucose, and then the glucose goes to the Krebs cycle, where it undergoes some more oxidation to release all of that energy in the glucose molecule. And finally, the byproducts of glycolysis and Krebs cycle go to the electron transport chain, which is able to produce ATP in bulk amount. So how does our body keep this blood glucose in such a narrow range and constant range in our body? So our body is able to do this differently depending on kind of what state the body is in. So the body can be either in the fed state, or our body can be in something that we call a fasted state. So you can imagine kind of the fed state as being just after you've eaten a meal. So let's say you've eaten a chocolate chip cookie. The glucose that has been broken up in your GI tract can then be used to directly contribute to this blood glucose level. And then, of course, the glucose can be used by our cells. Now, in the fasted state, so imagine that this is basically all the times you're not eating, the body has come up with kind of two different ways to regulate blood glucose levels. So now remember that in the fasting state, our body needs a way to essentially pump glucose into the blood to keep it at this level, essentially to replace the glucose that's being used up by our cells because we don't have this constant intake from, our chocolate chip cookie. So in this case, our body has glycogen, which is a polymer or kind of like essentially a string of glucose molecules that it essentially stores away. And our body actually kind of ingeniously makes this glycogen by using some of that glucose that is dumped into our body during the fed state. So kind of in anticipation of knowing that it's not always going to get glucose from eating, it kind of preserves some of it in this glycogen molecule. And most of this glycogen molecule is actually, just as a fun fact, located in your liver, which is why your liver is very, very important for carbohydrate metabolism. And so in times of fasting, our body can actually go ahead and break down this glycogen into the individual glucose molecules, which then can be used to keep our blood glucose levels constant. So unfortunately, it turns out that this mechanism of breaking down glycogen only lasts for about 10 to 18 hours in our body. That is to say that after 10 to 18 hours, we've essentially used up all of our glycogen stores, and we'd need to eat another meal to kind of build those glycogen stores back up. So you can imagine during an overnight fast, for example, so, when you go to sleep and, it's usually about, hopefully 8 to 10 hours. And so you can imagine there's a point during the day when your body needs another way of producing glucose. And so our body has come up with a second way called gluconeogenesis, which is indeed the topic of this video. And gluconeogenesis is exactly what its name implies. It is the genesis or creation of new glucose. Now, it's actually quite fascinating just to kind of think about this for a moment. What we're saying in gluconeogenesis is essentially our body is taking precursor molecules that are from a non-carbohydrate source. So essentially it looks at what it has lying around, and notably, most commonly it uses amino acids in our body, as well as a molecule called lactate, which is produced as kind of a byproduct, oftentimes in exercising muscle cells. And it takes kind of these precursor molecules and reconfigures them to produce glucose. And it's this glucose that can then be used to be dumped into our blood to maintain this constant blood glucose concentration and a constant supply of ATP for our tissues. So now that you kind of have a big picture of carbohydrate metabolism and where gluconeogenesis fits in, let's go ahead and talk about, as promised in this video, about this metabolic pathway, gluconeogenesis. So in order to do this, I think most effectively, it's actually important to revisit glycolysis briefly. So I'm going to go ahead and bring up the reaction diagram that was used to explain glycolysis in a previous video. So just to orient you, remember that glycolysis begins with glucose up here, and glucose is broken down in a series of steps. Most notably at one point it's broken down into this 3-carbon molecule, glyceraldehyde 3-phosphate, and then it is broken down even further and reconfigured, releasing some ATP and NADH along the way, and ultimately forming this molecule pyruvate. And pyruvate is a very important molecule because it can continue to the Krebs cycle, where it can be further oxidized to produce more NADH that can be used by the electron transport chain to produce ATP. All right, so that was kind of a big mouthful, but just remember, big picture, glycolysis breaking down glucose into pyruvate. Turns out, the way I like to think about gluconeogenesis is that the goal of gluconeogenesis is to produce glucose. And so essentially the way I like to think about it is that gluconeogenesis is almost the exact reverse pathway of glycolysis. That is to say, we start at this end of the reaction pathway. We start with pyruvate, and we essentially go funnel back the opposite direction through all of these reactions to produce glucose. Now, the key word is that it's almost the exact reverse of glycolysis, and it's almost the reverse because I want to call attention to these orange arrows. So note that there are kind of three orange arrows. So one from glucose to this molecule glucose 6-phosphate, another one here, and then one at the very end, which converts the last molecule to pyruvate. And what's important to note about these reactions in glycolysis is that unlike the other bidirectional black arrows that are used in most of the reactions, these orange arrows are unidirectional. What they're trying to indicate is that these three reactions are irreversible. In other words, they have a negative, if we pull out a fancy term from chemistry, they have a negative delta G value, or a negative Gibbs free energy, which means that if we were to reverse these particular reactions, we would have to flip the sign, right? And so these negative delta G values would become positive. And that's problematic, right? Because we know that in order for any biological reaction to occur, we must have a negative delta G value. So our body has come up with a bit of a compromise. Our body has essentially said, okay, look, we have 1, 2, 3, 4, 5, 6, 7 reactions with these kind of bidirectional black arrows that are essentially reversible. That is to say, they have a delta G value that's near 0. And so they can go either direction. So our body says, okay, we'll keep those 7 reactions. But in going from pyruvate back to glucose, we have to come up with a different reaction pathway for the 3 steps that are irreversible. So that's exactly what our body did, and in fact that's what I'll go ahead and review in the remainder of this video. But essentially you can say, with those 3 steps in mind, we're just performing the reverse of glycolysis.